Right, this is a copy of the mark scheme for the September CEDAR test. There are some questions in particular that I'm going to do sort of verbal feedback for as well, explain it in a bit more detail, how common mistakes people make, and some techniques that you might use to sort of improve. Question A and B, straightforward, knowledge. If we go down to question C, knowledge as well, but there are a few mistakes people made with this. The main ones being the idea that this one here, mistaking where the hydrogen bonds are. I'm not saying straight chains, you might say long chains, but the key bit is that the chains are straight and unbranched. So if we scroll up to the molecule to show you why those marks appear like that, so you can see that each of these lines is an individual cellulose molecule. Each one is a beta glucose, and the bonds holding them together are glycosidic bonds, not hydrogen bonds. The big mistake there. Glycosidic bonds holding the monomers together. So you've got the chains running in parallel to each other, and the chains are held in place by the hydrogen bonds, which are often shown as dotted lines. So the chains are held by hydrogen bonds, not the individual monosaccharides. And here's the table for question 2. Question 2, part B at the top. There are some other options apart from these, but they'll be in your notes. Things like long, large molecules, so it cannot exit the cell by diffusion, and insoluble, so it doesn't affect the water potential. Looking at question 3 and the graph, hopefully you can see that the line rises as temperature increases and then falls. The rate rises and then falls. You could also have said that the optimum temperature is 30 degrees. Big mistake was ignoring this part here, that the line didn't decrease, and then doing an explanation, so saying the enzymes denature. So you might say rate rises to 30 and then the enzymes denature. That is not a description, that's an explanation. So you only talk about denaturing enzymes for an explanation, explaining why the line falls. So this is a description. Looking at this question here, state maximum rate, and the key bit there is maximum. So you've got to find the part of the line where it's steepest, and that's between here. So to calculate a rate, you have to do the change in y divided by the change in x. So the idea that the mass change divided by the time change. So I've chosen within that part where the line is straight to go for this 5-minute interval here. So the time difference is 5 minutes. Go across with a ruler, go up and across with a ruler, and the difference between 400 and 200 is the change in y. So looking at this, difference in y is 200, divided by the difference in x, which is 5, to give an answer of 40. So that's the final answer there. There was 1 mark for just doing a mass divided by time. This question was particularly poorly answered, and it's one that does crop up time and time again, especially part A. So let's have a look at that. So for part A, so control group, why do you use control groups? It's to prove that whatever you're testing is having the effect. So in this case it's a drug, to prove the drug that's having the effect. It could be, if it's an enzyme investigation, to prove it's the enzyme that's having the effect, or it could be anything like that. Control groups are always treated exactly the same as the one you're doing the experiment with, in this case the people that are having the drug. And the second mark for this is always they're given something that doesn't contain the active ingredient that looks exactly the same. So if it's a drug, they're given a placebo. If it's an enzyme reaction, they'll use boiled or denatured enzyme instead to prove that that's what's having the enzyme's what's having the effect. So it's a standard answer, control group answer. Both parts, very, very common. So let's have a look at this graph before we move on to the last part where the mark scheme is. So I'll explain it here and then I'll show you the mark scheme after, because it's on a separate page. So the question, so if you look at group P, you can see that the line decreases. Now, most people said the line decreases for 4 hours, because that's after 4 hours the line does start to increase again. But the treatment wasn't given until 1 hour. So you can only say that the treatment decreases the volume of acid secreted for 3 hours, or between 1 and 4 hours. Big mistake. Make sure you make sure you look at when the treatment is being given, because most people will miss that, and it's a very, very common question. Here you can see that. That's the mark scheme. Question 5, part A and part B should have been very straightforward. For this question here, again, I'll go through the diagram and then I'll show you the mark scheme after. So we've got a situation where in tube A, we've got lactose. In tube B, we've had an enzyme which has broken the glycosidic bond in lactose. So now we've got two monosaccharides. We've actually got galactose and glucose. Hopefully you can see that there's more precipitate in tube B. So that indicates that you've actually got more reducing sugars in tube B. In fact, you've got two reducing sugars in B, galactose and glucose, and one reducing sugar in A, which is lactose. And here you can see the mark scheme for that. This last question, some very, well, not silly, easy mistakes to rectify, but a lot of poor exam technique in this. So it really illustrated people who've got good or poor exam technique at long answer questions. So it's breaking the question into two parts: structure and function of polysaccharides. So the structure of each polysaccharide and the function of each polysaccharide, not polysaccharides in general. So you should talk about starch, then glycogen, then cellulose. The mark scheme is on the next page. This is a copy of the mark scheme for the question. It might look a little bit complicated, but so if you started off with cellulose, so start off with cellulose, you get 1 mark for saying beta-glucose. Many beta-glucose are joined by condensation, and that forms glycosidic bonds between. You've then got a series of mark points which we've gone through before from the first question about the structure of cellulose, and then you've got 7 and 8, the function of cellulose. This one isn't, bottom one isn't always a marking point, number 7 is. For starch and glycogen, if you'd have said made from alpha glucose by condensation glycosidic bond again, you wouldn't get these 3 marks again. You only get that once. If you did it for cellulose, then you get those 3 marks for cellulose. You can't get them again for starch. So starch and glycogen, it's the same marking points. Insoluble, so it doesn't affect the water potential. Helical, which makes it compact, so it can store a lot of glucose in a small space. Large molecule, so the molecule can't leave the cell. And its function is it's a good store of glucose. You can only get a maximum of 3 marks for points 9 to 14. The idea being you can't then get 6 marks, so more than half the marks, just from one of the biological molecules. So that is the feedback from the September CEDAR assessment. Some of you will have been asked to take the quiz, which is also available on Moodle.